is actually quite important in the FLT3 data we're going to talk about later. But Philadelphia chromosome and CML I would consider a uniform mutation across all the malignant cells. That's not always the case in acute leukemias, as many of the mutations that occur are mixed in multiple clones that do or do not have the mutation. In order to target it or inhibit it, it ideally should be a gain-of-function mutation, meaning that the mutation confers an overactivity that you can suppress and ultimately prevent from the leukemia from growing. And ideally, it would be one that would increase the sensitivity of cells to non-chemotherapy-based treatments, as I think there's still very little way to improve what we give with real high-intensity chemotherapy. So I've listed a few down here that are, in my opinion, very successful strategies: Philadelphia chromosome, BCR-ABL targeting with CML, retinoic acid receptor alpha targeting with APL, and there's a number of new therapies for other cancers that I think really are improving those diseases. The one that I've spent a lot of my career working on is this FLT3 tyrosine kinase. It was originally described in acute leukemia in the early 90s. It's a self-surface receptor that's in normal physiology an important component of early T-cell development. There isn't clearly a lot of other roles for the FLT3 in normal myeloid cells, despite the fact that it's mutated often in acute leukemia. But it is mutated in about 30% of acute leukemia. Internal tandem duplications, which is a acquisition of a repeated portion of the protein in a particular region, occurs in about 25% of those leukemias. And then there are activating point mutations in what we call the tyrosine kinase domain, which occur in a much smaller fraction of leukemia. And if you look at what happened to patients who had these FLT3 ITD mutations historically, before the tyrosine kinases were in any real use, patients who had no mutant allele in these two Medical Research Council studies had a 5-year survival of about 42%. Based on how much FLT3 mutation that was found in the cancer really can influence how well patients do with treatment. So if you had a high level of FLT3, more than 50% in this paper, the 5-year survival was only 15%. So there's a dose-dependent effect, perhaps, that the more FLT3 mutation that's there, the worse the disease is, the more difficult it is to cure with chemotherapy alone. And I think that's been seen not only in this study, but in several others. So thinking of it as a target, you have to think of it as more than one particular target, because leukemia cells often have not only the mutated form of the tyrosine kinase, and either FLT3 ITD or tyrosine kinase domain mutation protein, but often also overexpress the wild-type kinase and can be dependent on that for signaling in certain subsets of leukemia. And that's important when you review the propensity of several of these drugs to do their job, which is blocking the signaling of FLT3. Some of the drugs, for instance, sorafenib, quizartinib, or AC220, don't do a very good job at suppressing tyrosine kinase domain mutations. The IC50s, the inhibitory concentrations required to inhibit kinase domain mutations with these drugs just aren't obtainable in human beings. Whereas some of the other drugs, like midostaurin, which was originally referred to as PKC412, crenolanib, gilteritinib, have the propensity to block FLT3 ITD and kinase domain mutations. So thinking about it as multiple targets is important when you think about how some of these drugs work and why they might fail. And for what it's worth, it's very typical for a patient when they do become resistant to sorafenib to acquire a tyrosine kinase domain mutation as a resistance mechanism, and sometimes that's treatable with some of the other drugs. We struggled for many years, and we talked about this a little bit at lunch, at trying to prove that any of these early drugs in development, FLT3 inhibitors, were very effective. And part of the problem was it was very difficult to find the right patient population or give it to a patient when they had relapsed or been treated several times and they really had no likelihood of responding to anything. And the early phase clinical trials with SEP701 or midostaurin showed mediocre responses, I would consider, and what I mean by that is many of these responses were just lowering of the blast percentage, not necessarily meeting any of the typical WHO CR criteria. And add to that, the doses that were given were often intolerable. Patients had a lot of vomiting or gastrointestinal complications, and as such, limited the ability to take them consistently. But what was learned from that was that we did see some link between the degree of inhibition of FLT3 when you looked at the leukemia cells after patients had taken their inhibitor and the degree of clinical response. And we also learned how to do an assay to determine whether patients were getting active drug in their system and doing what we wanted it to do. So I'm going to review here a little bit of more about the phase 2 study of midostaurin because I think it helps us learn a little bit more about why it may work in a combination setting. But this was a phase 2 study where they took 2 different doses of midostaurin and they randomly assigned it either 100 milligrams twice a day or 50 milligrams twice a day. And they were given to relapsed or refractory patients, but it wasn't specifically FLT3 mutated patients. Actually, 2/3 of the patients were non-FLT3 mutant or FLT3 wild type. And they had standard efficacy endpoints, but the results were quite disappointing. They had 1 patient in the whole cohort of 92 that had what met criteria for a PR, and the rest of the patients had blast reductions. But what was somewhat interesting was that the likelihood of developing of a blast reduction in this group of patients was significantly higher if you had a mutated FLT3 versus a wild type FLT3, and that led the investigators to believe that they had an active compound and wanted to move it on to the next phase with giving it with chemotherapy. And as you would expect, if you weren't seeing much classic remissions, you wouldn't expect any long-term survival, so the survival probabilities here are quite poor. And this is a theme that was not specific to midostaurin. Virtually all the drugs that are listed here, given as single agents as phase 1 or phase 2 studies, had mediocre responses up until we got to some newer generation drugs, which are fairly specific FLT3 inhibitors, quizartinib, crenolinib, and gilteritinib. And all 3 of those drugs showed remissions at reasonable rates, 40% in this study, 50% in the crenolinib study. But as I pointed out in earlier, they're still not achieving complete remissions by classic criteria, meaning the patients are not recovering counts. And when you looked closely at these patients on all these studies, there's still molecular evidence of leukemia. So it's, as single agents, in my opinion, still not a strategy that's likely to achieve any durable MRD negative remissions or cures, but it might be a strategy to maintain remissions or assist in getting an MRD negative state. there have been a number of reported combination studies now where we've taken what we've learned from the single agent studies and applied them to upfront therapy. We'll talk a bit about midostaurin and sorafenib studies. I will point out relapse refractory chemotherapy studies have been largely a failure, and I think that's not going to change with any of these newer drugs either. It's a harder disease to treat when it's relapsed or refractory, and I think the goal ultimately is not to let people relapse and do whatever we can to prevent that, because the survival after relapse has been poor all along. So I'm going to delve in a little bit more detail to the studies that were briefly mentioned by one of your colleagues. Sorafenib with chemotherapy study here was reported originally by Ravandi et al. To the JCO in 2010. This also was a study that did not require patients to have a FLT3 mutation, so it was an all-comers study. The idea at the time was a multi-kinase inhibitor, sorafenib, given with chemotherapy might improve outcomes based on cell cycle kinetics, and if they gave it with chemotherapy, they might see a survival benefit. And they randomly assigned patients to receive IA, which is their standard induction, plus or minus sorafenib. And as you can see here, there really wasn't much in the way of benefit from adding sorafenib to that treatment. And from their group, they decided that all comers was not the right strategy for FLT3 targeting or for sorafenib targeting leukemia. But they did a subset analysis and later published this information, which was that if you looked at the patients who had a FLT3 mutation in this study, there were a couple things that they learned. One is that they had a slightly higher rate of remission, 89% versus 74%. And they seemed to have a benefit up until the point where they stopped being treated. So there was maybe some indication that it was doing something to the FLT3 mutated patients. But it's only after they spent a little time looking at the molecular biology of the treated cells that they learned, I think, what caused that failure. So as we were doing these studies, we were perfecting or trying to perfect an assay in which we could take cells or take blood from patients and learn whether the drug was getting into their system real well. Basically what we've done is taken the plasma off, spun down blood, and this is then taken to a lab where the plasma is exposed to a FLT3 ITD cell line, and then you can determine whether that plasma has the ability to kill a cell line. And if it can kill a cell line, then the idea was that it could kill leukemia in the body. And if you looked at the Ravandi paper, what was reasonably clear was that at day 7, the FLT3 ITD was turned off completely in virtually all the patients, which is what you would want to see with a FLT3 ITD targeted agent. But what wasn't the case is if you looked at wild-type FLT3 in those same samples, it was still on. So the cells could acquire overexpression of FLT3 and survive the insults. And I would argue that's one of the reasons why that strategy didn't work, particularly in patients who had